Our last speaker today is Hyun, and he is a postdoctoral scientist in Dr. Thomas Lloyd's lab at the Johns Hopkins School of Medicine. He'll be speaking about disrupted organelle dynamics in autophago-lysosomal pathway in the Drosophila models of C9-ALS in vivo. Welcome.
Okay, I will share the screen. So can you all see this? Okay, I'm very happy to have this opportunity to share my study, which basically about the in vivo analysis in the Drosophila model of C9-ALS disease. In here, I basically trying to visualize and validate intracellular organelle dynamics so that we can better understand whether and how these organelle dynamics can be disrupted in these disease model. So the ALS is one type of human neurodegenerative disorder, which can be characterized by the specific degeneration of motor neurons. So the patients, they eventually display catastrophic loss of motor function. But beside of this, in the aspect of cellular physiology, the affected neurons also characterized by the mislocalization of protein aggregates, which suggests that there might be malfunction of protein homeostasis. Unfortunately, so far, the majority of the ALS cases are sporadic. However, it has been reported that several genes are associated with the familial form of ALS, and the G4C2 hexanucleotide repeat expression in the first intron of C9-AL gene is one of the cases of the familial form that we classified as the C9-ALS. And currently, there are three potential mechanisms in C9 pathology, which contains a loss of function and gain of function. But here, as we use the fly for our model system, we tested the gain of toxicity by expression of the G4C2 surrey repeat. So the main question was, when we focus on the first aspect, we can reasonably ask whether the organ dynamics of exon transport can be affected by the expression of the G4C2 surrey repeats, because motor neurons, they normally contain extremely long axon. In addition to this, when we focus on the second aspect, we can also raise the question that there might be disrupted organ dynamics relevant to auto-lysosomal dysfunction. So based on this, we build up our rationale and hypothesis that defective axon transport associated with auto-lysosomal dysfunction can be founded by the gain of toxicity in C9-ALS. So to validate this issue, we first mark out the lysosome for the key intracellular organelle in performing a protein degradation pathway. And here we see the axonal lysosome in fly motor neuron visualized by the lampone GFP. And as we can see here, the total number of axonal lysosome was not affected by the expression of the surrey repeats. However, we do see the signal aggregates and axonal lysosome, enlarged axonal lysosome by the expression of the third repeats, and these signals normally displayed very static movements. These are a representative live imaging of axon transport of a lysosome from the control and repeats expression animal. And actually, one of the hardness to quantify the axon transport of the lysosome in this system was they're very small and very fast. So to avoid these tracking difficulties, what I did was I classified these positive particles based on their signal intensity. So here we found that by the expression of the third repeats, we do see the increased aggregates, which normally displayed a high signal intensity and very static movements. You can realize that in the red dots is the number of the increase. And this is the quantification results. And as we can see here, by the expression of the third repeats, we do see the organelle accumulation and the increase the static population. During the retrograde transport, lysosome, they constantly receive the new membrane and membrane protein through the fusion with other organelles, such as autophagosome. So we also tested the axonal autophagosomal process in fly motor neuron as well. Very surprisingly, unlike the lysosome, we do see the less number of autophagosome in fly motor axon, but the remaining autophagosomes still display very secure retrograde movements. And this is the quantification. So here we see the reduced number of autophagosome and reduce the flux, specifically in retrogrades, but their speed and percent of movements was not changed, which indicates that the expression of the G4C2 may affect the axon transport of autophagosomal flux, but their movements is not disrupted. Because we know the neuronal autophagosome, they normally develop in the very distal part of the axon. So we want to ask, are the autophagosomes are accumulated in synaptic button? So we also check the synaptic button area as well. And this is the quantification result, not even in axon, but also in the synaptic button, we do see the reduced number of autophagosome. And this defective phenotype was very specific to autophagosome, because when we checked the mitochondria, the number of mitochondria was not disrupted, which suggests that there's the biogenesis of autophagosome presumably is downregulated in this animal. Because previously it has been demonstrated that the autophagy can be activated by the neuronal stimulation in synaptic button in fly motor neuron. So we applied the temperature sensitive TRPA1 channel to artificially upregulate the autophagy process. So when we see here in control animal, when we switch the animal from the 20 to 29, the number of autophagosome was upregulated. But this upregulated phenotype was still inhibited by the expression of the 30 repeats, which suggests that the biogenesis autophagosome may still inhibited under the 30R repeat expression. So the next step was we want to ask, how about the maturation? So there's a small number of autophagosome, but want to ask, how about the autophagosomal maturation? Because the autophagosome start to develop in the distal part, and they normally travel to retrograde. And during their retrograde transport, they can fuse with the late endosome to develop to amphysome, and fuse with the lysosome to develop to auto-lysosome. So we want to check the late endosomal axonal transport by using the GFP Rep7. And we do see the increased number of axonal late endosome and the disrupted axonal transport of this organelle. But very interestingly, the fusion process of autophagosome are still preserved. So not only the fusion with the late endosome, but also fusion with the lysosome, even under the expression of the third repeats, we can still see the very nice fusion process. So we want to ask whether these deficits causes from the global disruption of axon transport. So we want to check the other type of organelle. And one candidate is dense core vesicle, and the other one was the mitochondria. And this is the dense core vesicle movements by visualize the ANF GFP. And as we can see here, under the expression of the third repeats, we do see the increased number, which suggests the accumulation of the dense core vesicle. And also, we see the disrupted transport movements. But however, very surprisingly, in terms of the mitochondria, we don't see any disrupted phenotype. So this is the quantification from A is the number of the mitochondria per unit meter, and the B is the flux, and C is the percent of movements. But none of these parameters was affected by the expression of the 30 repeats. We also expressed the double repeats in second and third chromosome, but we still see the very nice movements, which suggests that the external transport of the dense core vesicle can be disrupted, but not mitochondria. And these indirectly represents that the defective phenotype that we found from the auto-lysosomal process is quite specific, and not because of the global disruption of external transport. So finally, we want to ask why there's some specific disruption of external transport to the auto-lysosomal process. And we want to focus on the ER dynamic because we know that the autophagosomal development is quite dependent to ER integrity. And there is some report that the lysosomal position is also regulated by the ER. So we check to the ER by using the signal visualization. And here is the time sequence imaging from the control, and this is the time sequence from the 30R repeats. And there are some ER tubule growth from the control, which from the ER tubule and the retraction. But these dynamic phenotypes was quite rarely discovered, observed under this condition. And another very significant difference was we do see the ER discontinuity and the fragmentation. So in summary, the expression of the G4C2 may disrupt the some specific organelle axonal transport, such as the late endosome and lysosome. And it seems likely that the 30 repeats generates a downregulation of the autophagosomal biogenesis. But however, the remaining autophagosome still undergo normal maturation and development process during their retrograde transports. And local accumulation of organelle, such as a dense core vesicle and late endosome, lysosome, associated with the autolysosomal process dysfunction is not because of the global disruption of axonal transport, because we do not see the disruption of mitochondria axonal transport. Rather, it may be need to consider the involvement of the ER integrity in these animal systems. I would like to appreciate the work in Lloyd Lab, and this work is supported by NIH R1 and Packard at Johns Hopkins. Thank you.
That was beautiful. I mean, those images are amazing.
Oh, so yeah.
Yeah, I was going to ask you. So just because I know very little about the Drosophila side of things, how hard is it? So these are live imaging that you're doing. Is that
yes, correct? Yes. So actually, we just select the third insta larvae right. Just before getting into the pupa, so which is the largest one, and then dissect from there, and then remove fat and intestine, and then just put in everything intact, and just so it is not easy, but when people practice several months, we can get used to that.
So it's like an ex vivo preparation, right? Everything else is intact, but you're still opening it up so that you're able to actually image the whole process. So
yes, exactly.
Very cool, very cool. So I don't know, this is maybe Rebecca has some question, because this is getting very close to the lysosomal and the ER type of things. But so I was curious about what is your future direction now that you're thinking more about the ER integrity side of it. would you be looking at ways to mark them? Do you start looking? How do you start looking at the ER integrity side?
So at this moment, the next step is we want to co-visualize the ER with other organelles. So whether for example, whether the autophagosome can co-localize and co-migrate together with the ER, that may directly suggest that the ER integrity is really required for the axonal lysosome, axonal autophagosomal positioning. So we are just looking at that.
Right. Yeah. No, that's very cool. Sorry, I didn't mean to put you on the spot, Rebecca.
I sort of do have a question actually, because my most recent project as for my master's was focused on C9-AL, where we were talking about phase separation and how that disrupts organelles at large. But I was kind of interested, probably this is not your area, but I was kind of interested to know how you think loss of function mechanisms relate to this, you know, considering that C9 is thought to be involved in autophagy itself. So how do you think that kind of feeds into your results?
Yes. So yeah, as you may realize that at the beginning, there are three potential mechanisms in C9. One is the loss of function, so haploinsufficiency of the protein level. Yeah, there's many report that the Oh, yes.
Sorry, just quickly, we're going to get cut off in like a minute. So I just wanted to thank you all because, you know, next session starts. So I'm sorry.